My name is Kate, and today we're introducing the qEV magnetic concentration kit, which uses functionalized magnetic particles to concentrate EVs quicker and easier than ever before. Before we get into the specifications of the kit, it is worth talking about why you might need to concentrate your EVs in the first place. This may be because you've started off with a dilute sample, such as conditioned media or a biofluid like urine. Alternatively, it could be that your EV isolation itself has resulted in dilution. The data from the literature shows that our qEV isolation columns result in purer EV isolates as compared to either precipitation or ultracentrifugation methodologies, whilst maintaining a good yield. However, even the best workflow may lead to EV isolates which are too dilute for some downstream applications, especially when measuring biomarkers. Currently, the most common post-isolation concentration method for smaller volumes, such as those used in diagnostics, are centrifugal concentration filters and our own non-magnetic qEV concentration kit. Both are okay for most small-scale lab studies, but their time-consuming 75 to 90-minute protocols and the need for centrifugation in both makes them unscalable and difficult to automate. It is for these reasons that we developed a new solution, and this is where the qEV magnetic concentration kit comes into its own. This technique employs magnetic nanotrap extracellular vesicle particles, which were produced for us by Ceres Nanosciences to gently concentrate intact EVs. Nanotrap particles are composed of hydrogel polymers that have been functionalized with chemical affinity baits. We optimized the kit to drastically reduce the incubation time to just 10 minutes, resulting in a protocol length of just 20 minutes from start to finish. This makes it around 4 times faster than other methods. And that's all well and good, but faster is no good if the concentrating doesn't work well. Well, to reassure you that it does, we did our own little pilot study. We split our starting sample in 2, then isolated both using qEV original columns. We then pooled these 2 isolates and split them back up again. One we kept as it was, and the other went through the qEV magnetic concentration kit protocol. We then resuspended the pellet from the qEV magnetic concentration kit in the same volume that we took from the non-concentrated sample. This meant that we had the same volume and could compare the degree of concentration before and after the kit had been used. For the comparison, we turned to qPCR, isolating RNA using the qEV RNA extraction kit, and then using a qPCR assay for miR16. As you can see, the levels of miR16 were significantly increased in the samples which had been concentrated using the qEV magnetic concentration kit. This increase in detectability could be the difference between a valuable EV biomarker being detected or not. The actual degree of concentration is variable and is controlled by you. This is because the qEV magnetic concentration kit leaves you with a pellet, and the volume in which the pellet is resuspended has a large bearing on the degree of concentration. For protein or RNA analysis, the most concentrated samples can be achieved by directly resuspending in the lysis buffer of choice, preventing unwanted dilution. It is worth noting here that the qEV magnetic concentration kit is not suitable for functional studies, as the EV magnetic nanotrap capture particles bind irreversibly to the EVs. However, the qEV magnetic concentration kit is suitable for downstream analysis of nucleic acids, for example, PCR or RNA sequencing, and proteins, such as Western blots or mass spectrometry. This versatility for diagnostic-style research and testing, coupled with the swift and automatable protocol, makes the qEV magnetic concentration kit an ideal partner for diagnostic applications. If you're ready to accelerate your EV concentration protocol, follow the links below.